do the whole lecture over again?
No. That's one reason why it's better to actually attend the class than to try to view videos, is there's going to be a couple of lapses. Okay, anyway, so we're talking about CIRM, California Institute for Regenerative Medicine, and we're talking about stem cells. And what we're saying is that, you know, stem cell initiatives, stem cells are a big deal. And it's, how does that feed into medicine? We're going to provide replacement parts for people, basically. Cells that go back into the body. Our bodies are made out of cells. There's a problem, we can fix it by adding back some cells. That's the idea. Okay, so that's interesting that there's this website. If you go up to the top here, it says about stem cells. That seems like a pretty good place. Stem cell basics, stem cell definitions. Click that. Okay, so definitions. What is an embryonic stem cell? What is a pluripotent stem cell? An adult stem cell, an iPS cell, a cancer stem cell. Right? But we've touched on that a little bit already. There's a little bit of confusion. It's like, well, there's a stem cell, and then there's a couple different flavors of stem cell. So the first one is, what is an embryonic stem cell? Embryonic stem cells exist only at the earliest stages of embryonic development. They go on to form all the cells of the body. They no longer exist about 5 days after development. So that means the fertilized egg, as it's forming a blastocyst, you know, that it keeps dividing and then forms this ball. And one of the first things that occur is that there's some cells, some very special cells inside the blastocyst that are going to give rise to all of the differentiated cells within that new embryo. And it's called an inner cell mass. So the inner cell mass is just a group of cells, just within the blastosphere. And it's just in one little corner, and if you tease out those cells, those are embryonic stem cells. So that's kind of easy. Embryonic stem cells come from the embryo. Can they come from somewhere else? No. Then they wouldn't be embryonic stem cells, right? So that the embryonic stem cells come from the embryo. So, you know, that's not such a big jump. You kind of think, well, fertilized egg, yeah, it's going to give rise to a person, right? Or an animal, or, you know, whatever it's designed to do. So that means all the cells from the body must come from those beginning cells. That kind of makes sense. It's no big jump. So it's not surprising that there would be cells. You could isolate some cells, and they would have the potential to form these different things. And I think that it's that intuition that everyone has that makes us a little bit unimpressed sometimes with what the scientists are talking about. It's like, oh, well, we pulled some cells out of an embryo, and look, it's beating like a heart. And it's like, well, yeah, you destroyed this like living creature that was going to become not just a bunch of throbbing cells, but like a person. And, you know, and so what? So you got it in a dish, and it's made less than a heart. It's made some kind of just a little grouping of cells or something, and it's beating. So I'm not that impressed with that. So, but, you know, I mean, what's impressive is sort of what nature designed there. The fact that all the cells of the body are going to be derived from this one cell type. That's the miracle. And it's not really the scientist that's got it in the dish. It's like, yeah, you've created something less than what was there originally. So I feel a little embarrassed by that sometimes, by their enthusiasm. But anyway, it is powerful, and it's powerful by nature's design, not by what's been done by getting into a dish. But anyway, so that would be potentially a great source, but on the other hand, it's a little bit of a problem ethically, right? It's like, well, now we don't have people, but we have a couple of dishes floating around. But there's a huge excess, right? There are lots of embryos that don't become people. Fertility clinics, a lot of embryos. Currently, there's like 400,000 embryos in fertility clinics being stored. How many people were derived from fertility treatments, like what they call IVF, in vitro fertilization? What's the number in the world's population, do you think? I mean, it's obviously more than one, more many people do you think have been created for in vitro fertilization? 20%? No, that's not that high. Maybe in Northern California it might be, but not in the world at large. So it's about 5 million people. So 5 million people were created through in vitro fertilization. So that's a technique. It's like, wow, that, it didn't, maybe, we personally were not created that way, or maybe we were. No big deal either way. But, you start to think, well, some of this biology stuff, this is kind of significant. There's 5 million people walking around because biologists can manipulate some of the cells involved in fertilization, and couples that couldn't give birth naturally or whatever, that they can assist that process, and that's led to 5 million successful births. Pretty big deal. So one of the guys that's involved with that is an Australian guy, Alan Trounsem. And Trounsem is now the director, I think he's still the director of CERN. So they brought him in. But anyway, I don't know if, how long that will last per se, but anyway, so that's the embryonic stem cell stuff. But a pluripotent stem cell, what's meant by that? Pluripotent. Well, potent means, mighty, able to do something, has strength, fertility somehow, potent. Pluripotent means many. So it's able to produce everything. So a pluripotent stem cell just means it's able to produce all the cells of the body. Is an embryonic stem cell pluripotent? Yeah, right? I mean, that's the kind of the most powerful one is the embryonic stem cell, because we know that can give rise to everything. So by definition, it is pluripotent. Then there's the adult stem cell. Well, that means that you can isolate cells from us. We're all adults. Actually, right after birth, biologically, you're considered an adult. So we're all adults. So there's fetal and then adult from a biology perspective. So an adult stem cell is a stem cell derived from an adult. And so where do these cells exist? Is there an inner cell mass somewhere in your body? No. The inner cell mass lasts for about 5 days, a couple of days, inside the blastocyst. That's a step in the formation of an embryo. So with the adult stem cells, there's pools of these things. So there's some in your bone marrow, adult stem cells. So those, the bone marrow is responsible for forming a lot of the blood cell types. And so these cells keep going, they keep pumping out blood cell types. But they themselves are not blood cell types, they're stem cells. And they reside in the bone marrow, and then they differentiate, they bud off, and they give rise to some of these differentiated cell types, but they remain as a pool. Same thing in the skin, actually. There's some stem cells in your skin. And also, pretty much in every epithelia in your body, there's stem cells. In the skin, there are about 1 every 20. Like if you look at a 2D cross-section of skin, and you're looking at the basal epithelia, so right at the junction between the epidermis and the dermis. So that's called a basement membrane zone, so that interface. So right at that interface, there are basal cells right on top of that are epithelial. And in like 1 out of 20, if you look at them in 2D, you see, like, stem cell, not stem cell, not, 20, another stem cell. Well, these things are pretty potent, so they can give rise to daughter cells that then rapidly divide. They themselves don't divide that rapidly, but they give rise to very rapidly dividing cells. So that way they're not pressured to divide that much. Why is that a good thing? To not divide too much. Every time you divide, there's a chance for errors. I don't know, you guys photocopy stuff sometimes, like you photocopy the notes. I miss class, I photocopy the notes. It's kind of low-tech approach, but a higher tech approaches go on YouTube and watch this. But anyway, so you photocopy notes. And then somebody said, hey, I missed it too, can I have your notes? Oh, I photocopied these from Matt. It's like, oh, okay, but So, they might have been photocopied a couple times. Each time they're photocopied, the copy gets kind of grittier and grittier. There's errors, there's a couple of stray points or whatever, and then next thing you know, it's full of little dots and stuff. So, copying is a problem. When DNA replicates, it's got to replicate a pretty substantially sized genome, and there could be errors in the base pairs. So there are various errors that are possible, even large-scale errors. Chromosomal breaks are even possible. So there's lots of problems with copying. So stem cells aren't being copied. So adult stem cells are in the body. There are kind of pools that allow your body to keep replenishing needed cells. Something's got to give rise to everything, and so these adult stem cells are there. They're not embryonic, though, and they're not pluripotent. So the stem cells that are in your skin, they can give rise to more of these epithelial cells. Maybe you could tease them, in culture to do a couple of other things related to skin. But, they're not going to form like maybe some other cell types very easily. Maybe they wouldn't form a liver cell all that easily. So there is some level of commitment of differentiation, but so they're not pluripotent, but they have some significant potency. And in an IPS cell, it's an induced pluripotent cell. Well, this is a human construct. IPS cells don't exist unless they're created in the lab. Okay, so embryonic stem cells, that's part of normal reproduction, normal creation of an embryo. Adult stem cells, those are some cells, pools of cells in your body that help you maintain cell populations. IPS cells, induced pluripotent cells, scientific invention where we can add a couple of genes to cells. I think there's typically around 4 different genes that are added. Mostly transcription factor type things, OCT4, SOX2. And these 4 factors can give rise to a cell that that functions like an embryonic stem cell. Wow, that's a pretty big invention. It's like, yeah, that is a big invention, and that means that we don't really need to go back to the embryonic stem cell. So it means like that controversy that society has about deriving embryonic cells could be partially relieved by the IPS cell technology. Nonetheless, whenever some human manipulation occurs, we quite don't have control over everything. You ever notice that? Sometimes it goes well, sometimes it doesn't, which is why we all sort of watch as the space flight goes off, where everyone's like hoping for the best, but every once in a while they crash and burn. But anyway. So IPS cells, yeah, you can do that, and actually in mice, they can take these cells and they can grow an embryo. They can reverse a cell. So like an IPS cell could be any cell, like a skin cell. Skin cells are easy to get, right? And we work with them. So we can take a skin cell in the lab and put in transcription factor genes and derive a IPS cell which functions like an embryonic stem cell. That was implanted in the uterus, give birth to a new creature. So in mice, that works. Isn't that amazing? You can take a skin cell from a mouse, add a couple of genes, and plant those cells into the uterus. You give rise to a litter of mice that were derived from a skin cell. There's no embryo there. There's no reproductive. Big deal, right? It's like, wow. Gee, didn't understand that well. So then you can sort of see how deep the stem cell thing is going. It's like, you can do all these weird things. Cancer stem cells. Let's go down to what their explanation is in that. So there's a lot of money spent on cancer research. So, you always hear about cancer, and it's because we spend billions of dollars every year on cancer research, and for decades we've been spending a lot of money on cancer research. So that's why it comes up all the time too, and it is a common disease. But there's maybe a subpopulation of some of the cancer cells that are self-renewing, and that the idea is that you could call these a stem cell. So there's kind of a core to the tumor, and that what they've observed is that if they add drugs to stop stop the tumors, like chemotherapy for this sort of thing. They kill a lot of the cells in the tumor, but then it seems like there's always some cells that escape. They escape the chemotherapy, then they metastasize somewhere else in the body. Next thing you know, there's another tumor. And it's like, well, why were those cells able to escape? Well, they were perhaps cancer stem cells. It's like they eluded the cancer therapy because they weren't dividing that rapidly. And one of the things about stem cells is they don't divide very rapidly. They give rise to cells that divide super rapidly. So that's kind of the idea. So they themselves don't replicate that much, but they represent the store of potential for cancer tumor. So it's cancer stem cells. Okay. So I mean, the good thing about this is it's online. You should look at this stuff, and this is in your backyard. And you live in California, and California committed $3 billion to this stuff. I mean, you know, and I mean, this is changing your world. You know, you drive by and you see, oh, look at this campus. Wow, where'd they get the money for that building? You know, it's like they got it from CERN. I mean, a big chunk of it, other donors, etc. But I mean, CERN is putting a lot of money into infrastructure too. So that kind of demystifies some of what's going on. Let's see. On the right here, there are some Flickr images. I thought these were kind of nice. Pictures of cells. So, like in our lab, we're looking at cells all the time. You've already looked in the microscope and seen some cells. And we use this technique called IHC. So, immunohistochemistry. And it's a way of getting color to localize to particular cells. They use antibody reagents. So antibodies recognize a protein, and that protein is specific to the cell. So certain cells they know carry a certain protein. Fibroblasts, they have actin. You know, smooth muscle fibroblasts, they have smooth muscle actin. You know, you can have an antibody that recognizes that protein, it's going to go to the fibroblasts. Is it going to go to the epithelial cells? No. Epithelial cells are not like muscle cells, right? Fibroblasts are like muscle cells. So you can get specific labeling. So they use that technique over and over again. So you see these colorful pictures of cells everywhere. You know, whenever you see articles on this stuff, they're showing you pictures that look like this. Immunohistochemistry. We do that all the time in our lab. We've got that big microscope there. It's got a very bright light source that then runs through some prisms, and it separates out very defined wavelengths. So these, basically they're filters. And so then you're stimulating with one wavelength, and then a different wavelength is given off. And that's the fluorescence idea. We'll talk about that more when we get to that in class. But what I wanted you to realize is that other people are looking at cells besides us. And this is getting to be like mainstream stuff. So you're thinking, well, I'm in this lab and it's, you know, just sort of one of, you know, just some little side thing. This is not a side thing. This is like $3 billion of Californians' money. It represents a major research effort. You know, the IVF thing has contributed, you know, 5 million people to the planet. I mean, you know, this is a juggernaut. This is moving ahead. And you know, there are jobs in this stuff, and there's things to be done. And you know, you guys are a new generation, so I mean, what are you going to do with this stuff? I don't know, you know. But it's powerful and it's useful. And you know, as is typical of human endeavor, usually when you start with something, you think, oh, we don't need this, and then a couple of years later, it's like, we really need this. This is a big deal. So you know, turn of the century analogy is Westinghouse and Edison saying, hey, you know, we're going to run like wires that carry current into the city of Niagara, New York, because there's a Niagara Falls there, and we can have these generators that will, you know, they're like paddle wheels, and the water's running over them, the paddle wheel's turning, and then as it turns, it's generating electricity. We're going to take these wires that have the electricity, we're going to run them throughout the city. It was the first city that was electrified, I think, in the world. right? That's pretty impressive. So forward-thinking people. It's like, and a lot of people spoke up. It's like, we don't need no damn electricity in our city. What are you guys doing? You know, I mean, we know that electricity is dangerous. You can get zapped, shocked by electricity. So we don't want it. We've got horses. You know, we can get from place to place. If it's dark outside, we light a candle or a lantern. It's all good, right? Yeah, but the lanterns fall over. The fuel starts a fire, burns down the house. The candles, they don't burn that long. They're not very bright. The wind blows. They blow out. I mean, it's a lot of problems. So electricity might really help. So they put in electricity, and now it's like, well, the power goes out. Like with the recent storm in New Orleans, it's like, well, there's almost 1 million people without power. That's tough. That's really a tough thing. But yeah, but 100 years ago, that was thought, oh, we would never want to have power there, right? So it's sort of typical of the whole human thing, right? So you're thinking, well, why do we need stem cells? You're going to find probably, I don't know why, but you're going to find you probably really need this. So anyway, so they have some cells that are differentiated from the embryonic stem cells. Endocrine cells, pancreatic cells. Pancreatic cells are kind of a cool thing. Common disease that involves pancreatic cells. What? Yeah, it's always that. But something to do with insulin. Diabetes, yeah. Okay, so type 1 diabetes there's not insulin being produced. So the cells in the pancreas that are responsible for insulin production have been damaged. You could replace those, take some stem cells, differentiate them into the insulin producing cells, put them back. These people are no longer diabetic. Well, that's kind of cool. And it beats, having to inject yourself all the time and monitoring your glucose levels and stuff. That's a real, potential for a cure. So you can see, well, that's pretty exciting, yeah. So that's some of the kinds of research that are going on, and you should look at all these pictures. There's just a lot of them, and a lot of them look a lot like the sort of pictures that we generate in our lab. There's pictures of cells. These blue dots all the time, those are nuclei. The mountain that we use for our cell preps has got DAPI in it, and DAPI is a chemical that binds to DNA, and it glows blue. So you see these blue dots, those represent the nuclei in all these pictures. We do the same thing. So they show also that you can derive heart cells and various things here. And then they show you a couple of buildings too. It's like, wow. Stem cell center. UC Irvine. That's one example. It's like a nice building. This is a little bit closer to home. The Lokey Stem Cell Research Building, Stanford. So, you guys ever visited Stanford campus? I worked at the Stanford Medical School for several years. So these guys build a lot of buildings. There's a lot of money at Stanford. But they got some summer money to, which also supplemented a gift that they had from a very wealthy university that acquired Lokey. But CERN put in another $50 million, so now they've got this big stem cell center. So anyway, I encourage you to take a look at this sort of stuff and get excited about cells and what you can do with cells. This class is not specifically on stem cells, and a lot of the stuff we do actually, we haven't worked with stem cells on some of the things, but we're constructing groups of cells, and it doesn't matter if where they come from. So they can come from stem cells. So that's actually the weak point in the whole stem cell thing. Remember, I was telling you that when we first were able to culture cells, people were thinking we're going to put these back in people, and it doesn't work, right? So even to the current day, with iPS cells and everything else, they're thinking, well, we can put these back, and is that working? Not really. I've got some money invested in some stocks that are trying to do this. Companies like Advanced Cell Technology is one of them. They have retinal epithelial cells, and that's a common form of blindness when those cells are no longer functioning correctly. So you could add the pigmented retinal epithelial cells back to the eye, and then if they could reside there, then vision could be restored. It's like, well, nothing better than making the blind see, right? That's kind of a convincing selling point for stem cells. But it's been a couple of years. They're still trying it. And actually, you talked to one of the guys at the Buck, Deepak, and he's got his project is on those cells, and he shows you some data. It's like, yeah, those cells, they repopulate the back of the eye and stuff. It's like, yeah, but if they really did, then people would be helped right now. So you might have one result that shows that, but show me that you do it 50 times in a row, that works every time, and it's actually doing something functional. So we're not quite at that point yet. So there's some problems with cell assembly and getting cells to actually be able to reside in the body and be functional, right? Well, that's exactly what we do. So we're working on that stuff. So I think it could fit in really well with stem cells. Okay, so one other thing I wanted to show you, so there's CIRM. So that's the local California thing. But California is part of the United States. Right? Remember those guys in Washington? Where the Republicans, where are they right now? They're in Tampa, right? Are they in Tampa, their convention? Anyway, so there's the rest of the United States. What are they doing? Well, the main organization involved with health in the United States is who? What's the institution that is overseeing health in America, your well-being? The big Uncle Sam. Who is it? For your health. Who? Yep. National Institutes, plural, of Health. So it's this big organization located outside of Washington in Bethesda, Maryland. And so it's NIH, National Institutes of Health. So I'm writing the address here, stemcells.nih.gov/research /NIH research SC unit, stem cell unit. Huh. Okay, maybe I don't need it. Okay. So I didn't need it. Okay. So this is a page from the National Institutes of Health. I would encourage you to look at that. That's a huge resource. Oh my gosh. Like NIH.gov. It's part of your government. It's what you're paying for. They're in charge of overseeing your health. They fund education. So the NIH paid for me to get a PhD. Right? So do you guys know that, by the way, that you can get a PhD for nothing? Still, pretty much? Huh? Yeah. So you didn't know about the master's program. Do you guys know about the SURE program this summer? That Brandy was in our lab this summer, and she had a paid job for 8 weeks, and was it 8 weeks or 10 weeks? 8 weeks. 8 weeks. She was able to learn a bunch of stuff in the lab. It's a summer job, and you can put that on your med school application and stuff. You guys don't know about that, and then you don't know that there's a master's program. And we were saying that Dominican college, if you have only undergraduate programs, it's college. And once you have advanced degrees, it's university. So one of the advanced degrees offered at Dominican is a master's degree in biology. And so that's one of the reasons why it's Dominican University, right? So, and this master's program is done in conjunction with the Buck Institute and also BioMarin and us. So we're providing students in all kinds of research environments. So, and then there's, so master's degree, and then there's the PhD. And if you get into PhD programs, a lot of times the labs are funded through the National Institutes of Health. So, the principal investigator, a guy like me, or a girl like Maggie, or whoever, we write these grants, and the NIH might decide to fund that, and then we've got a few $100,000 typically, but could go higher into the millions to do research. And then we buy a bunch of lab equipment, and we start giving people jobs, and so the NIH really drives the whole thing. And when you get into PhD programs, a lot of times your principal investigators got an NIH grant, and so they're using your labor to learn some things in the lab, so they pay you, and so it's kind of a wash on what your education costs are. So that's not a bad deal. Also, medical school, you can get an MD, PhD. You guys know about that? So a lot of people are interested in an MD degree. I want to be a doctor, but you could also get a PhD degree. It's like, well, I don't want to be in school forever. But actually, a PhD often takes 5 to 6 years to do, and a MD PhD often takes no more than 5. It's like, wow, I can get both degrees in possibly less time than chicks that just get a PhD, yeah. And on top of that, you're probably going to go to medical school for free. Wow, that's pretty cool. You guys don't know about that, right? Care about medicine, you're thinking, I want to be a doctor, but I think it's going to be making too much money. You could go for free if you were in an MD, PhD program. Because again, it's funded research stuff, big buildings being built. Drive down 101, you see the Buck Institute, a lot of that money is coming from CERN. I was impressed that they built a new wing to the building, and that was CERN money. Now I saw online that there's more construction that's going to be going on, and that is also CERN money. I mean, it hasn't even been built yet. So the whole place is going through this metamorphosis. It's going to be a big deal. Okay, so this is just a stem cell part of the NIH website. That I encourage you to look at the whole NIH website, but go to this specific address, stemcells.nih.gov, and then you can also do this part if you want, and then get into the whole stem cell stuff. So there's a lot more to human health than stem cells. Nonetheless, stem cells are up there, and it's being actively pursued. There's protocols online. Wow. Okay, so this is a lab course. I'm giving a lecture, but I'm trying to pull you guys in, trying to have you relate to this whole thing. So we're not just playing around in some funny back room lab somewhere. It's like we also have we're doing experiments, and we have protocols, and we can go online and find protocols. They describe how to do experiments. Basically, they tell you that you need to incubate for 24 hours with the medium, etc. Just like the protocols you're dealing with. So the stuff that you're dealing with is, a lot of these protocols and stuff. You can find them online. You can find them off the NIH website online. And who are they? It's like the government is in charge of your health, and they fund this with billions of dollars every year. And they're interested in having this happen. That they oversee things. They provide a lot of protocols and stuff. Do they invent everything? No, they invent a little bit maybe. From their perspective, they probably think they invent a lot, but they're mostly funding things. So the real research comes from a lot from the university labs and some from the company labs. So I just wanted to give you that perspective. It's kind of interesting because the guy on this particular protocol, this is senior author Ron McKay. He hired me years ago, Cold Spring Harbor Laboratory. We never got along too well, but I survived nonetheless. Some of the things he's done are brilliant, and some of them are kind of weird, but that's Ron. So anyway, he's definitely made a contribution, and that kind of points out too. It's like that's the way it is for scientists. they're people too. So they do some amazing things and some non so particularly amazing things. Did you guys ever hear of Linus Pauling? He won a Nobel Prize for the structure of like alpha helices, like collagen forms an alpha helix, so protein structures. And he was the main competitor on the structure of DNA, so Watson and Crick DNA structure. So he came up with the same kind of molecule, but he had the base pairs on the outside. Watson and Crick had a face this way. That was the difference between the two models. So, but anyway, Linus Pauling had won a Nobel Prize already, and he had an institute in Palo Alto, and he would hang out down there too. But later in his life, he got really into vitamin C. Did you hear about how important vitamin C is? So he was this guy talking about everybody needs mega doses of vitamin C, which I'm not really sure that that's true. He kind of went overboard maybe on vitamin C. It's important, but maybe not in mega doses, etc. So okay. All right. So now I want to transition into what it is that we're doing now. Now I find that I should have been on that page, but maybe I don't need to deal with that. Growth curves were on the NIH stem cell page. It's like, so you mean the experiment we're doing today, we're going to play around with some cell numbers? I mean, that's important enough to be in the NIH stem cell page? It's like, yeah. I thought we were just doing some little funny thing with the cell counting thing and all that. No big deal. Big deal. Stem cells, you're going to need to understand how they grow. What their dynamics are. How often do they divide? And so there's a population doubling rate. So if you draw that out, you're going to get a curve. So you believe me, right? I can go back to the page and click on whatever on the side that shows you the growth curves to that, but let's just draw it out. If you want to know how to get there, by the way, under stem cell information at that site that we were on, stemcells.nih.gov, there's just growth curves off to the side like that, and it shows you a bunch of different growth curves. Okay. So this is a lab class. We got to get into the lab, but I'm trying to get you interested in what we're doing. Point out that this stuff is pretty significant, and you're at the cutting edge. So not only are you taking a college class, but you're at the cutting edge. Not wonderful. Okay, so the basic thing with a growth curve is you're going to have cell number and you're going to have some amount of time, like days. And the cell number is going to be increasing over time. So at 0 time, let's say we start out with 10 to the 4th cells, and then later we end up, we go to 10 to the 5th cells, 10 to the 6th cells. So you're going to get a line. It's a line if over here, if this is a log plot. You guys know about exponents and log? What that means? So it's exponential growth. So, if you take, it's doubling, right? So if you take 2, things are doubling. And then to the first power is equal to 2. And 2 to the second power is 2 times 2. It's equal to 4. And 2 to the third power, third generation of doubling, is 2 times 2 times 2. It's 8. Right? So, this comes up in biology, exponential growth, logarithmic. It's like almost the definition of logarithmic growth. It's one of the drivers to this whole interest mathematically in exponents and log. So log of cell number. You've seen the log plots too. That like this, it's like an uneven hash marks. Because yeah, that's the way the log works. So anyway, but if you plot it on log on one axis, but not a log plot on the other, so that's why it's a semi-log plot. It's not the log of days, but the log of cell number. Because it's double. 2. First generation, second generation, third generation. You got a line. So that's what we're kind of embarking on today. We're interested in creating growth curves. So we've got some cells growing. They're dermal fibroblasts. Just sort of a garden variety cell, but that cell could be made into an induced pluripotent cell, which could give rise to a whole new organism. Right? So, and typically, with the stem cell stuff, a lot is done with skin cells. You read some of the articles, look on the website and stuff. Well, we took skin cells, some of those pictures on Flickr that I was showing you, and we clicked on Flickr on the right side of the NIH stem cell page, and we were looking at pictures. A lot of those pictures show skin cells that were made into some other sort of cell type. First made into the induced pluripotent cell, and then cultured in a particular way on gelatin with some additional nutrient or whatever, and next thing you know, it's a liver cell. Next thing you know, it's some other cell type. So skin cells are a big source of cells because they're so easily accessible. Okay, so you expect some sort of doubling there. Is this going to go on forever in a dish? I mean, how far can the cells grow in a dish? Right? We're always what we call splitting the cells, moving them from one dish to another dish. Why do we have to move them from one dish to another? Why can't we keep them on one dish? It's like you're fishing in a fish tank. Why can't you leave the fish in the fish tank? Yeah, there's not enough room. The cells are dividing, right? So they divide, and remember I used the word monolayer. They're creating a monolayer. And I said that's a scientific term, monolayer. And that means that the cells are right next to each other, but they don't grow on top of each other too easily. Especially primary cells from your body don't do that. Cancer cells sometimes pile up on top of each other. Primary cells are contact inhibited, so they go to a monolayer. Okay, so if you don't move them from the dish, then there's going to be some limitation to the growth. It's getting crowded on the dish. Kind of reminds you of planet Earth. At some point, population has got to plateau out. So this is a plateau here. And we can call this the growth phase. The cells are really growing logarithmically. How about at the beginning of this whole thing? When you first sprinkle some cells onto a dish, do you expect them to take off right away? No. They need an adjustment period. It's kind of like you guys waking up in the morning, right? The alarm clock goes off at 6:30, whatever. You're like, you're not ready for class yet. So things are kind of delayed at the beginning. It's a little bit of a delay for the growth really starts to take off. So this delay here, call that the lag. So there's a lag phase, a growth phase, and a plateau, or stationary phase. So all cells in culture do this kind of thing, right? The classic way of doing the growth